Hello, in this video I'd like to explain the fundamentals of needle EMG. This video is intended for neurology trainees and therefore requires some knowledge of basic neuroanatomy and physiology. This is what it looks like when we perform a needle EMG. We stick a needle in a muscle and record these strange looking squiggly lines on the screen while mumbling inexplicable phrases like polyphasia and long duration motor unit potentials or a reduced interference pattern. In this video, I'll try to explain the underlying processes going on in the muscle that result in the signal as we see it on the screen in healthy subjects, in patients with nerve lesions, and in patients with myopathy. So what do we actually look at when we do an EMG? Well, to properly understand the EMG, we must begin by refreshing our knowledge of the way our muscles work. All skeletal muscles are activated by nerve cells called motor neurons. Each motor neuron is connected to a number of muscle fibers, and all muscle fibers connected to the same motor neuron are part of the same motor unit. If you make a cross-section of the muscle along this dotted line, you would see all muscle fibers alongside each other like this. When an action potential travels down the nerve, all muscle fibers belonging to the motor unit will depolarize and contract simultaneously, as indicated by the yellow color here. Muscles are innervated by a large number of motor units that lie interspersed within the muscle, each with a territory of about 3 to 10 millimeters. It's important to realize that the size of the motor unit varies substantially between muscles. Smaller muscles that require very precise control, such as eye or hand muscles, have smaller motor units, whereas less sophisticated muscles requiring less subtle commands can do with much larger motor units. For example, eye muscle, among the smallest and most subtle muscles we have motor units as small as 25 fibers, whereas a motor unit in a biceps can encompass several thousand muscle fibers. What we measure when we stick a recording needle in the muscle is the electrical activity of motor units. The concentric needle electrode has a recording surface of about 0.5 millimeter. This means that only a handful of all fibers belonging to the same motor unit will contribute to the motor unit potential, the MUP. Fibers closer to the needle will have a larger contribution than fibers further away. When we see a MUP, we can measure a number of things: its duration, its amplitude, and the number of phases. And for these parameters, we broadly have the following reference values: no longer than 50 milliseconds, no larger than 2 millivolts, and no more than 4 phases. In the following slides, I will show how these parameters are affected by neurological diseases. In patients with a partial loss of motor neurons, for instance with polyneuropathy or partial nerve injuries, the first thing that will happen is that the loss of a particular motor neuron will lead to some muscle fibers being denervated. After about 2 weeks, these fibers will start producing spontaneous electrical activity. This low amplitude activity appears on the screen as fibrillation potentials and positive sharp waves. As a metaphor, think of it as small children who have lost their mother. They start crying out for help. This activity is often referred to as denervation activity, although personally I prefer not to use that term in this context. Although it's true that this activity is only caused by muscle fibers that have lost contact with their motor neuron, this activity can also occur with patients with a myopathy, and as such the phrase denervation can be somewhat confusing. To avoid confusion, I prefer to use the term spontaneous muscle fiber activity. In subject with mild injury, some motor neurons will not be affected, and in the initial stages you would expect the remaining motor unit potentials to therefore maintain their normal morphology. As time goes by, something remarkable happens. Due to the regenerative ability of the peripheral nervous system, remaining motor neurons start to sprout and start to reinnervate these muscle fibers. To keep with the analogy, the mother of a neighboring family has heard the crying muscle fibers and offers them to join her family. In the early stages of reinnervation, these sprouting nerve fibers are not fully mature and they're not yet fully myelinated. This means that these newly reinnervated muscle fibers depolarize slightly later, causing a polyphasic motor unit potential. As even more time goes by, these newly formed sprouts mature and all muscle fibers now fire simultaneously again. This reinnervation process has led to an increase in the density of muscle fibers belonging to the same motor unit, leading to a MUP with a higher amplitude and a longer duration. Note that the amplitude of the MUP is also strongly influenced by the position of the needle relative to the motor unit, whereas the duration is not. This is why MUP duration is considered to be a more reliable indicator of prior nerve injury than amplitude. Although personally I feel that both can be evaluated reliably, and amplitudes are slightly easier to judge in a live recording. Nonetheless, probably best to look at both parameters. After a complete nerve injury, we will again see spontaneous fiber activity, and in this case it can be quite abundant. Remember that it takes generally 2 weeks for this activity to occur, so it's recommended to wait for at least 2 weeks before performing an EMG in patients with suspected nerve injury. In this case, due to the complete loss of motor units, no MUPs will be seen. This is what we call a loss of continuity, and this is an important finding, as it may suggest the presence of a severe nerve injury that may require surgical intervention. Once nerve fibers start to regenerate, for instance after a surgical intervention, at some point a regenerating axon will enter the muscle and will start forming new connections. Initially, these axons will innervate only a small number of muscle fibers, and nerve endings will be quite immature, resulting in small polyphasic motor units. This can often be seen before the patient has noticed any return of muscle function themselves, and therefore they are a hopeful sign that clinical recovery is just around the corner. As the reinnervation process continues, similar to what we saw in partial nerve injuries, MUP amplitudes and durations will continue to increase until we see this large amplitude, long duration motor unit potentials as we've seen before. So what happens when we stick a needle in a myopathic muscle? Here the situation is quite different. There is no loss of motor units. Instead, the muscle fibers themselves are sick and therefore less functional. This leads to a number of things. Some muscle fibers will die, resulting in smaller motor units. Some fibers will have trouble generating an action potential along the membrane, and nerve conduction along the membrane may also be disturbed. The end result is a smaller, short duration, polyphasic motor unit potential. Important to note is that in some myopathies, for instance inclusion body myositis, muscle fiber splitting and muscle fiber regeneration do occur, leading to the presence of muscle fibers that are not connected to an axon. As we've seen previously, these muscle fibers will also start producing spontaneous muscle fiber activity. So that concludes my explanation of MUP morphology. However, to be able to interpret the EMG, we will have to answer one last question about electrical muscle activity, which is, how do we regulate the amount of force we apply? When we apply very little force, we start out by activating a very small number of motor neurons at a very low firing frequency. As we apply more force, two things happen. The first motor neuron starts firing more rapidly, and another motor neuron joins in, usually at a slower rate. The minimum rate at which a motor unit can fire is approximately 5 Hz, and for every 5 Hz increase, another motor unit joins in. This process is called recruitment. So what would that look like in our schematic overview? Well, here you see a motor unit consisting of green muscle fibers, and these are all firing at a very slow rate of approximately 5 Hz. When more force is applied, two things happen. The firing rate of the green motor neurons increases to 10 Hz, and another motor unit, in this case the red team, becomes active and starts firing at 5 Hz. Unfortunately, our current equipment does not allow us to see these different motor units color-coded. As a clinical neurophysiologist, I really hope it would, but it's a bit more difficult than that, because what we see is the black line. And part of being an experienced clinical neurophysiologist is being able to look at this trace and be able to gain an impression of how many motor units are active. When the patient applies even more force, even more motor units become active. In this case, the blue ones join in. For this presentation, I haven't tried to draw a black line that sums up the activities of these 3 lines, because it would be a bit complicated, but you'll appreciate that it becomes real messy real quick. If a healthy subject applies maximal force, there are so many motor units contributing at such a high rate that they're no longer recognizable as such, and the whole line just becomes a blur. This is what we call the interference pattern. So what happens in patients with a severe nerve injury? Well, in this case, I'll show you a very extreme example. And there's only one motor unit left near the needle and all other motor units have been lost. As the patient attempts to apply more force, the motor unit starts to increase its firing rate. But as it looks around to see whether their friends are joining, the motor unit realizes that there's no one there. This leads to an increased firing rate, but no recruitment. And this reduced recruitment pattern is something typical of a severe nerve injury. In myopathies, things are quite different. As I've mentioned before, the number of motor units is not reduced in patients with myopathy. However, the number of healthy muscle fibers, and thus the amount of force that each motor unit can generate, is reduced. So what happens here is that as the patient tries to apply more force, there's an initial realization that the force delivered by the initial motor unit is insufficient, and immediately all its friends start to help out and become active as well. and this is a phenomenon that we call enhanced recruitment. This can lead to a very blurry trace, as you can see here, in which individual multi-unit potentials are barely recognizable. So in summary, when we do an EMG, we interpret it by looking at several parameters. First, we look for the presence of spontaneous muscle fiber activity. Then we try to evaluate the multi-unit potential and its various properties like amplitude, duration, and morphology. And then we look at the interference pattern as the patient applies more force. And altogether, these things are useful for us to gain an impression of whether this is a healthy muscle, a neuropathic muscle, or a myopathic muscle. I hope that this short overview has helped you gain a better understanding of how we interpret the EMG. But I'm also aware that things will all of a sudden look a lot more difficult to interpret when you're looking at a live trace. In the future, in the next video, perhaps I'll show you real examples of various conditions to show how these principles apply to actual data. To give you a brief first impression before I finish, here is a live trace. If you focus on the left-hand side of the screen, you'll see that the height of the screen is about 2 millivolts, which means that any multi-unit potential larger than the screen is enlarged. On the x-axis, you'll see that the total distance is 200 milliseconds, which means that the distance between two of these tiny dots is 20 milliseconds, and any multi-unit potential longer than these two dots is prolonged. Now, if you look at this video, try to see if you can judge the recruitment pattern as well. Can you tell whether this EMG is normal, neuropathic, or myopathic? Let me know what you think in the comments, and I'll soon be back with more real examples and explanations. So that's all for now. Thanks again for watching. Hope you enjoyed this video. Let me know in the comments if you have any questions at all. And don't forget to press the like button if you appreciate the video. Hope to be back soon with new material. Bye.